The topic of the meeting today is Spinraza and Zolgensma for spinal muscular atrophy, effectiveness and value. I'd like to begin by asking the members of CEPAC to introduce themselves.
Good morning. My name is Dr. Jason Wasfi. I'm a cardiologist at Mass General Hospital. I'm an assistant professor of medicine at Harvard Medical School, and I'm one of the medical directors of the Physicians Organization, and I'm a vice chair of CEPAC.
Good morning. Good morning. Stacy Brown. I'm associate professor at the University of Connecticut School of Medicine and also the director of the Community-Based Education Office.
I'm Austin Fracht. I'm a health economist. I'm also the director of the Partnered Evidence-Based Policy Resource Center at VA Boston Healthcare System, and I have affiliations at Boston University School of Public Health and the Harvard School of Public Health.
Good morning, I'm Steve Kogut. I'm a professor at the College of Pharmacy at the University of Rhode Island, where I direct our graduate program in health outcomes research, and I'm a member of the Rhode Island Drug Utilization Review Board for Medicaid.
Good morning, my name is Stephanie Nichols. I'm a psychiatric and substance use disorder pharmacist and an associate professor at the Hudson University School of Pharmacy. I'm also affiliated with the Tufts University School of Medicine.
I'm Brian O'Sullivan. I'm a pediatric pulmonologist at Dartmouth-Hitchcock Medical Center and professor of pediatrics at the Geisel School of Medicine at Dartmouth College.
Hi, I'm Reverend Albert Whitaker, formerly with the American Diabetes Association and currently doing consulting work.
Jean Ryer, I'm director of the New Hampshire Citizens Health Initiative at the Institute for Health Policy and Practice at the University of New Hampshire.
I'm Claudia Gruss. I'm a gastroenterologist with Western Connecticut Health Network, and I also have appointments with University of Vermont and Yale University.
Good morning. I'm Martha Gold. I'm a senior scholar at the New York Academy of Medicine and professor emerita at the CUNY Medical School.
I'm Chris Jones, network director of ventures at the University of Vermont Health Network and a trained health economist.
Good morning, Kim Lenz. I'm the clinical pharmacy manager for the Massachusetts Medicaid program at the Office of Clinical Affairs, which is a division of the University of Massachusetts Medical School. And I'm also assistant professor in family medicine and community health.
Thanks. And I'm Rob Azeltine. I'm professor and chair of the Division of Behavioral Sciences and Community Health at UConn Health and director of the Center for Population Health there. At the beginning of every New England CEPAC meeting, we want to ensure that all speakers disclose any financial relationships with industry. Every member of our New England CEPAC panel who is present today has met the ICER conflict of interest policy. We would also like to confirm that all of our members have no new conflicts to disclose. Seeing none, I'll now turn and ask that our clinical experts and patient representatives introduce themselves, and also would you please disclose any conflicts you might have with the health industry.
Hi, I'm David Michelson. I'm a child neurologist at the University of Loma Linda. I do a neuromuscular clinic, and I have no conflicts of interest to disclose.
My name is Emma Cefaloni. I'm a professor of neurology, pediatrics, and OBGYN at the University of Rochester. I've been specializing in neuromuscular diseases pretty much my entire career, and I treat neuromuscular diseases across the lifespan from adults and pediatrics. And I run Cure SMA clinic, MDA clinic, and Duchenne clinic. So my conflict of interest specific to SMA include the fact that I am a member of the DSMB for the AveXis trials. I also participate in advisory boards for both Biogen and AveXis, among others company. I do not have any financial interest in neither company. And I also receive grant support from Cure SMA for our multidisciplinary SMA clinic.
My name is Brandy Akins, and I'm a patient advocate. I have a son that had type 1 SMA. He died right after his second birthday, and I'm just here to advocate for our population.
Hi, I'm Danielle Sun. I'm also a mother of 2 children affected by SMA. My daughter Ruby is affected with type 3, no longer ambulatory, and my son Landon is diagnosed with type 2. I'm a board member for Cure SMA and have done other patient advocacy advisory panels like this for Avexis and Biogen in the past, but nothing ongoing.
Thank you. Throughout the day, we'll have other speakers and roundtable participants, and we will also ask them to disclose any conflicts of interest that they might have. I'd now like to introduce Dr. Steve Pearson, President of ICER.
Good morning. Thank you very much for being here today. And that goes especially for our patient representatives and our clinical experts who've traveled to be with us today. We really appreciate your being here for many reasons. And I know the audience will appreciate their participation. And thank you for being here with us. I just want to go through a few aspects of today's meeting that I think are important to put it into context. You've heard who the members of the New England CEPAC are. I need to tell you a little bit more about ICER, which is the academic, if you will, secretariat, and we host and convene these meetings. ICER is an independent, not-for-profit research institute based here in Boston. And this is our updated funding chart that shows that the work for the reports and for the meetings like today come from nonprofit foundations, 77% of our overall revenue. And that comes chiefly from several different foundations, among which are the Laura and John Arnold Foundation, the California Healthcare Foundation, the Blue Shield of California Foundation, the Kaiser East Bay Community Foundation, and several others. We do take revenue separately for what we call a policy summit program, in which we convene leaders of the life science industry with payers and PBMs and work with them around evidence policy looking forward. And those funds, as you can see, come a little bit more from the pharmaceutical side than the payer side, but it's used for those policy summit programs only and not for the research or meetings like today. So why are we here today? I think that's probably self-evident, but I would like to start with quotes that we heard from patients and their families. So bear with me, I know they're a little bit long. Here's one. The first year of my son's life was full of tears, 911 calls, and constant fear of losing him. SMA would rob our son of many things, including his ability to swallow, breathe on his own, move, and smile. He had surgery at 3 months old for a G-tube, as he could no longer safely eat orally. At 7 months old, he had a tracheostomy, as he could no longer breathe on his own, and we'd nearly lost him more times than any person should ever even consider. Another quote. I know that realistically I'll probably never walk. Regardless, having a treatment that could give me more strength opens up so many possibilities that would drastically improve my quality of life. If I could go to the bathroom by myself, roll over in bed, unbuckle my own seatbelt, or hold a baby, then I could travel anywhere I want, never have to wake up anyone at night for assistance, drive to work by myself, and change my future child's diaper. These may sound like menial tasks to some, but to me it would be a dream come true. So what you have here is both the burden, if you will, of the disease and the potential benefits of treatment, which are both really remarkable in this situation. So that's why we feel a certain special responsibility about our work today on this report and about the meeting today. Now, we're here for several reasons. What happens the day treatments like Spinraza or Zolgensma, assuming it does reach FDA approval, what happens the day that they're approved by the FDA? Well, the first thing you can imagine is that the families and the patients, like those that I quoted, rejoice. After years of sometimes their own involvement in the clinical trials, their own help and support to raise and focus the research, there's new hope. There are new options. And there's a heroism to the science and the determination that goes into bringing these treatments through clinical trials to the bedside. There is absolutely no doubt about that. What also happens the day these treatments are approved by the FDA or thereabout is that the company will set a price. And I need to be clear, there are no costs. There's a price that is consciously set by a company. Now, sometimes there are competitive issues, sometimes there aren't. If a drug is the first of its kind, if it's the first to be able to help a condition, there really isn't competition. There's a decision made about what the price is. And after that, insurers and employers who've been watching it maybe, or maybe haven't been watching it for months, will in a sense make some early decisions about how it will be covered and about how they may or may not be able to pay for it over time. And there are lots of discussions with the manufacturer. Sometimes things really don't get going until 3 to 6 months after FDA approval. But that process begins that same day or even before. And that's why meeting now, even though it's early days for some of these treatments, makes sense because a lot of really important decisions are made. So the historical context is also, I usually give a little bit of a speech at the end. I'm going to steal some of my time up front just to set this context more. I mean, it's interesting to think it wasn't always this way. Up until the early 1900s, patent medicines were considered highly unethical for some company to, or even an individual doctor to say, I've got something and it's a secret and I can sell it, but you can't know what it is or copy it. That was literally banned by the medical societies of the United States. I know that because my great-great-great-grandfather was expelled from the Western Medical Association for hiding the fact that he had figured out how to bring quinine west of the Mississippi. And he made a buttload of money. And his reputation went down the tubes. That changed when chemists in Germany, Bayer and others, started actually doing real science. And instead of kind of taking natural products, made different kinds of treatments. And behind the science later came investment because it took money to be able to put together the trials that eventually started to be required. And out of that came a different approach to patents and a different sense of what was right and wrong in bringing new treatments into clinical practice. Interestingly, around that same time, and certainly in the post-World War II era, insurance got started because all of a sudden healthcare was recognized as becoming so expensive that most people really couldn't afford it. And instead of just paying the doctor with chickens or whatever, it was going to be a business. So we had patents, we had manufacturers, we had insurers, we had costs going up, and we had the sense that we face new challenges, certainly in the era going forward. One way to view that challenge is to think about where we are with SMA, right? It's an ultra-rare condition, and we have 2 treatments that we'll talk about today. But view that as a point, and there's going to be concentric circles. The next one out might be other rare diseases. So there are some estimates that as many as 10% of Americans have an unknown, if you will, or known rare disease that they could have treatment for. And we've had meetings just in the past 6 months on several of those, like familial amyloidosis, hereditary angioedema. So there are lots of other rare diseases. Outside the rare disease circle, you've got a circle of all of our other healthcare needs. Just this past week, another new drug was approved for major depression. You might have seen it, a drug called esketamine. A very interesting drug, new mechanism of action, very much unmet need, patients who haven't received successful benefits from other treatments. The early estimates are that there are 5 million Americans who could benefit from this treatment, and it's been priced at launch at $35,000 a year. 5 million Americans times $35,000 a year is $175 billion. It's not going to be that much. Not all patients are going to use it. But that's another circle. We've got SMA, other rare diseases, other healthcare needs and costs. And there's a circle beyond that, which is outside the healthcare system. There are other societal needs and goals. There's education. There's trying to take care of the environment. There's the defense. There are other social needs. We are always going to feel the challenge of the balance between these different circles because we can focus in on any one place, but there are concentric circles of need and of cost and of trade-offs that have to be made. So that's one of the reasons that we're here today. Because ultimately we need to find a balance, and it's not an easy balance. To find the right balance, we have to start by drawing as much as we can from the experience of patients and their families and the clinical experts who care for them, trying to understand what it really is like to live with these conditions and what the true benefits of potential treatment are from their eyes. And that's, again, why we're so pleased that you're able to be with us today and why we really want to thank the patient groups who worked with us throughout the whole process. A balance is going to force us to be as clear as possible about where the evidence is and where it isn't, where it's clear that things work and where we might have questions, and how we factor that into our decision making and making some kind of balance out of this equation. And it also involves us looking at costs, or prices again, because price is the conscious decision that leads to the cost. And because ultimately, none of us could afford these drugs without insurance, or very few of us could. And insurance means that we share those benefits, but it means we also share a responsibility to try to work together to find that balance. And so I think the real reason we're here today is because we feel that it's better to do that in public, talking together, looking at the evidence, hearing from those involved, and wrestling with these issues. It's better than having it done behind closed doors. It's better than having it done when no one really understands why or what's happening. So it's not perfect, it's messy, but we are going to try to be as honest and as explicit as we can as we try to create this balance together. So I thank you again. Let's bring this process into the light and see what we can do together. With that, again, and with the thanks, let me tell you more about how the report was structured and brought to this process. So it started out almost 8 months ago with a scoping phase where we reached out and got guidance from patients and clinical experts, the manufacturers themselves, and other stakeholders about where the real questions lay and what we should know before we got started. Then our staff did the analysis of the evidence. We always reach out to see if there are patient-reported outcomes. We reach out to see if the companies have evidence that might not yet be in the public domain that we can learn from. Along with that, we do a cost-effectiveness analysis, and today we are privileged to have 2 researchers from the University of Sheffield who perform that work with us and will be here to present its results. We go through several rounds of public comment and revision, and we do have expert report reviewers. For this one, it was Catherine Kundrat, who cares for patients with SMA, has done so for many years. And how is the evidence and other material in our reports structured, in a sense, to lead up to the discussion and voting for today? Well, first, we identify a specific goal for all of our work together, and that is the basic idea that we want to seek a fair price, fair access, and future innovation. And to get there, we think many policymakers really need to think about 2 aspects: the long-term value for money or the short-term affordability. Now, long-term value for money, which we spend a lot of time talking about and voting on, is really a composite itself of 4 different dimensions. One is a look at the comparative clinical effectiveness information. We do then also have an incremental cost-effectiveness analysis, looking at the long-term cost-effectiveness. We'll spend a fair amount of time talking and voting on 2 other aspects of long-term value for money, and they are other potential benefits or disadvantages, things that might not be captured by the clinical trials or captured by a cost-effectiveness analysis, but which are important to considering value. Similarly, contextual considerations are always there somewhere. They can be a variety of different aspects about the condition, its severity. It can be aspects of other treatments on the horizon, but it's something that will be part of people's overall judgment. And for short-term affordability, we provide in our reports a potential budget impact analysis that allows policymakers to kind of make their own assessment of what the uptake of these treatments might be and what its potential impact on the budget would be at a national level. So that's the way that the information, and in some ways the whole day is scripted. It's to help us put together these pieces of information and to try to express our questions and our judgments in a way that can hopefully lend more transparency and a sense of explicit judgment to a process that can seem obviously very complicated. So the overall agenda is to catch up from my going on too long and to start with the presentation of the evidence. Following that, we will have a phase of manufacturer public comment and discussion, followed by other public comment and discussion for the CEPAC. We'll break for a relatively early lunch and then reconvene for further deliberation and votes. Then following a very short break, we will start about an hour and 15 minutes of a policy roundtable where we will be joined, where our patient representatives and clinical experts will be joined by 2 representatives from the insurance world and also 2 representatives, sorry, from the manufacturers. And it'll be during that conversation where we'll talk about how the evidence and judgments and uncertainties flow through, if you will, into coverage policy, into aspects around future research, pricing considerations, and obviously clinical practice. So we hope very much that we'll get through that and have a final round of reflections from the CEPAC members before we adjourn by 4 o'clock. So thank you again for being here, and why don't we start? I do have the information here about the clinical experts who have already introduced themselves and the patient experts. So let's start with our evidence review from Dr. Alexandra Ellis from ICER, who will present to you the review of the comparative clinical effectiveness.
Thank you. I'd like to first acknowledge my collaborators, Kristen Mickle and Serena Heron-Smith, and we have no conflicts of interest to disclose. So spinal muscular atrophy, or SMA, is a rare genetic disease, and it affects part of the nervous system that controls muscle movement. In individuals with SMA, both copies of a gene called SMN1 have mutations. And SMN1 is responsible for producing an essential protein, survival motor neuron protein, or SMN protein. And this protein is essential for the health of motor neurons. Individuals with SMA also have at least one copy of another gene called SMN2. And SMN2 produces a small amount of functional SMN protein, but not enough to compensate for the lack of protein generated from SMN1. But because it does produce some amount of functional protein, the number of SMN2 copies is associated with the severity of SMA. So again, individuals with SMA have an insufficient amount of SMN protein, and without this protein, motor neurons are lost. And this loss can lead to progressive muscle weakness, which can cause difficulty with moving, breathing, and swallowing. Historically, diagnosis was prompted by the clinical signs and symptoms of muscle weakness. And diagnosis is confirmed by a genetic test that looks for mutations in SMN1. More recently, there's been more pre-symptomatic screening of infants to identify and to diagnose SMA sooner, perhaps before symptoms occur. And so within the past year, SMA was recommended as a condition for which to screen all newborns in the United States. And since then, many states have adopted universal screening for SMA. But to focus on symptomatic cases, historically, SMA has been categorized into types, as listed here. There's also type 4 SMA, which is a very rare and mild form of SMA that presents in adults. But we're going to be focusing on types 1, 2, and 3. In the second column, you can see the distribution of SMA cases by type. And type is determined in part by age of onset, as well as motor functioning, so in the third and fourth columns. And type 1 SMA is the most common and severe form of SMA. So individuals present within 6 months of age, and they are unable to sit independently. Muscles in the respiratory and digestive tracts are affected, which causes difficulty with breathing and swallowing. And in the absence of treatment, individuals with type 1 SMA may die within 2 years of life. Types 2 and 3 SMA make up about 40% of SMA cases. And collectively, we're going to refer to types 2 and 3 as later onset SMA. These individuals present after 6 months of age and are typically less severe compared to those with type 1 SMA. However, they do still experience progressive muscle weakness over time and may need assistance with mobility. So for standard of care consists of a multidisciplinary approach that includes help with breathing and nutrition as well as other approaches, with the overall goal of to improve the health of an individual. So in type 1 SMA, that can include support with breathing and swallowing, as well as palliative care. And also there may be constant care that's needed to help prolong survival. In later onset SMA, it may be more mild, but they may still need assistance with mobility. So for example, patients may not be able to move their arms or legs and may need help with the wheelchair. But for all types of SMA, individuals are typically reliant on family members and other caregivers for this supportive care. And supportive care is not treating the underlying disease. And broadly, the goal of disease-modifying treatments is to increase the amount of SMN protein. And there's been a variety of approaches that have been explored to do this. So Spinraza is the first and currently only approved treatment for SMA. And it was approved in December 2016. Spinraza works by increasing the amount of functional protein generated from SMN2. It's administered as a lumbar puncture into the spinal fluid, the fluid surrounding the spinal cord. And there's initial 4 loading doses, and then treatment is needed every 4 months afterwards. Zolgensma is a novel therapy currently being reviewed by the FDA, with a decision expected by May of this year. Zolgensma is a gene therapy, and it works by delivering a functional copy of the defective SMN1 gene to the motor neurons. It's a one-time intravenous injection. And there's been considerable excitement in the community about both of these treatments, because as disease-modifying treatments, they have the potential to alter the natural history and treatment of SMA. And we heard from many caregivers how they often feel helpless and fearful, while also needing to be vigilant and providing constant care to a loved one. We heard how difficult it is to watch the disease progress in a child. And many individuals commented on the importance of early diagnosis and treatment. Patients and caregivers reported wanting treatments that improve strength and the ability to live independently. Many of these comments also highlighted the importance of not only improved strength, but also retaining and stabilization of current motor skills. And in adults with SMA, we heard how frustrating it is that these interventions have not been studied and that more data were needed in this population. So the scope of our review is individuals with SMA of any age and any type. Again, we're looking at Spinraza and Zolgensma, each versus supportive care. And the key outcomes that we're going to focus on today are survival, permanent ventilation, motor functioning and milestones, and harms. So focusing on Spinraza first, we identified studies in type 1 SMA, types 2 and 3, and also pre-symptomatic SMA. And today we're going to be focusing on these 3 trials listed here. There are additional smaller studies and observational studies, and the results from those studies were generally similar to the results in these trials, and so you can find the details of those studies in the full report. So in type 1 SMA, the trial had 121 infants that were randomized to Spinraza or to sham. So these infants had symptoms occurring before 6 months of age. They all had 2 SMN2 copies, and the mean age at treatment was about 5 months. The trial terminated early after positive results from an interim analysis, and at that time, about 64% of patients had at least 6 months of follow-up. And children are invited to participate in a longer-term study. Here are the results in terms of survival and permanent ventilation. So there was about a 47% reduction in death or permanent ventilation among patients on Spinraza versus sham control. About 84% of patients on Spinraza were alive versus 61% on sham. And 78% were not using permanent ventilation on Spinraza versus 68% on sham. And here are the results in terms of motor milestones achieved. So as you can see, none of the patients on sham achieved any of the motor milestones listed here. Whereas those on Spinraza, about 22% achieved head control, 10% rolling over, 8% sitting unassisted, and 1% standing with assistance. And in longer-term follow-up, about 45% of infants who had continued using Spinraza achieved full head control, and 29% achieved sitting independently. In later onset SMA, there was a trial with 126 children that were randomized to Spinraza or to sham. And the age range of these children was between 2 and 9 years, and they all had symptoms presenting after 6 months of age. And just note at baseline, all of these children could sit without support, but they could not walk independently. This trial also terminated early after positive results from an interim analysis. And at that time, all children had 6 months of follow-up, and about 43% had completed a 15-month assessment. And here are the key results for the later onset trial. In terms of survival and permanent ventilation, there were no deaths reported in either the Spinraza or sham arm, and permanent ventilation was not reported. And again, this is something that's not common in this population. For motor milestones, about 20% of the children on Spinraza achieved at least one new motor milestone versus 6% of those on sham. The differences were not statistically significant. But in looking at the motor functioning as measured by a scale called HFMSE, the average score improved among those that were on Spinraza, whereas it declined among those that were taking sham. And so this magnitude of benefit is something that's quite uncommon in the absence of treatment. Looking at pre-symptomatic SMA, so this is a single-arm study in which 25 infants were enrolled. They all had genetically confirmed SMA. 15 of the children had 2 SMN2 copies, which is predictive of type 1 SMA. And 10 of the children had 3 SMN2 copies, which is predictive of later onset SMA. This is an ongoing trial, and only interim results have been presented at conference abstracts. And at the most recent interim analysis, the median age of the children was 26 months, and all have been followed up for at least 15 months. And here are the results in terms of the pre-symptomatic population. So at the most recent interim analysis, all children were alive, and none were on permanent ventilation. There were 4 children with 2 SMN2 copies that required some respiratory intervention after an acute illness, but this was not considered permanent ventilation. And additionally, many of the children achieved motor milestones. So all of the children could sit independently, and 68% of the children could walk alone. In terms of harms, across all of the trials, discontinuations were very rare. In the type 1 study, the discontinuations that occurred from adverse events all had fatal outcomes, but this was not considered treatment related. The lumbar puncture procedures were generally well tolerated in all of the trials. Specific adverse events related to the lumbar puncture procedure were reported in the later onset studies. And in the trials of infants, the participants were nonverbal, so these types of outcomes are probably not fully captured there. And finally, beyond the clinical trial data, there were 2 additional potential safety concerns noted in the Spinraza prescribing information. So the risk of thrombocytopenia and potential for kidney damage. Switching now to Zolgensma, the evidence base currently consists of 1 non-randomized study, and that's in infant onset SMA. So this study had a low dose and a high dose cohort, and we're going to focus on the results from the 12 infants that were in the high dose cohort. So all children had 2 SMN2 copies, and the mean age at treatment was 3.4 months. I just want to know that these infants were diagnosed and treated earlier than those in the Spinraza type 1 study. So children who are diagnosed, children who are diagnosed, hold on one second. It's restarting.
Windows update. Windows decided it would be a good time to update right now. So we're just going to switch this out. Not for long. That's what you're doing. No, you have it too.
Slide.
There we go.
Where were we? Zolgensma. So I think I was just commenting on how the infants enrolled in this trial were diagnosed and treated younger than those in the Spinraza type 1 study. So children who are diagnosed earlier may be more severe, but earlier treatment may have greater benefits in terms of survival and gains in motor functioning. So it's difficult to compare these results between the trials because of the difference in the enrolled populations. And these infants were followed up for a total of up to 24 months, and there's been longer-term data as well. So in terms of the key results from the trials, all 12 infants were alive and none are on permanent ventilation after 24 months of follow-up. And many of these children achieved motor milestones. So 11 had full head control, 9 rolling over, 10 sitting for 10 seconds, and 2 standing and walking. And in longer-term follow-up, an additional 2 children were able to stand with assistance. In terms of harms, there were a couple instances of liver inflammation. So the first child who received a low dose of Zolgensma had elevated aminotransferase levels, and this led to a protocol amendment in which all patients received oral prednisolone. And then in the high-dose cohort, there was one patient that required additional treatment with prednisolone, and 2 children had elevated aminotransferase levels but did not require additional treatment. And so based on this clinical evidence, there are some remaining uncertainties and controversies. So first, the eligibility criteria of the trials meant that the enrolled populations were more homogeneous than you would see in a clinical practice setting. So some of the children that weren't enrolled may be more severely ill or have comorbidities like scoliosis. And so the magnitude of results that we see from these trials may not extend to those settings. The trials indicate that earlier treatments may lead to greater benefits, but there's currently only limited data on the pre-symptomatic SMA population. So again, just these single-arm trial of Spinraza, and only interim results are available right now. The trials were generally short in duration, and the Spinraza trials did terminate early. So there is some remaining uncertainty in terms of the longer-term durability and potential harms for these interventions. But to date, there hasn't been any waning of effect observed, which is promising. And again, these studies are still ongoing. Because of the differences in the enrolled populations that I mentioned in the Spinraza and Zolgensma type 1 studies, there is some remaining uncertainty regarding the comparative effectiveness of these two interventions. And finally, there's an open question regarding the combination or sequential therapy of these drugs. So some of the children in the Zolgensma study went on to take Spinraza after the 24 months follow-up, and some of these children achieved additional motor milestones. And so it's difficult to interpret what these results mean. So here is our evidence ratings for Spinraza and Zolgensma by type of SMA. So for Spinraza, we rated the evidence as A in type 1 SMA because of the large magnitude of benefits observed in the trials. In later onset and pre-symptomatic SMA, we rated the evidence as B+, in part because we had more uncertainty in these populations compared with type 1. So in later onset, there was more uncertainty because we saw benefits in terms of improvements in a motor functioning scale, but not in terms of survival or motor milestones achieved. And in pre-symptomatic, again, it was an ongoing trial, and there's only interim results presented at conferences. For Zolgensma, we rated the evidence as A in type 1. So this was a single-arm study with a small population, but it was felt that because of the magnitude of benefits in these 12 patients was so large, that there was considerable certainty that there was a high benefit of treatment with Zolgensma compared with standard of care. And we rated the evidence as an I, so insufficient to distinguish between Spinraza and Zolgensma in type 1 SMA. And we rated the evidence as insufficient for all other populations and comparisons due to lack of data. And beyond the clinical trial evidence, we heard from patients how there are other potential benefits and items to be considered when looking at this evidence. So the full report has a more comprehensive list of these items, but I'm just going to highlight a couple of the key points here. First, Spinraza is the first approved treatment for SMA, and Zolgensma is currently being reviewed. And if approved, a one-time intravenous injection with Zolgensma may reduce patient and caregiver burden compared with repeated lumbar punctures with Spinraza. An effective treatment with either intervention can lead to improvements in motor functioning, and that can allow patients greater independence. Children may be able to participate more in school, and caregivers may be able to return to work. And children may also be able to participate more in other community activities. And again, these are the types of benefits that are not being captured in the clinical trials. In terms of public comments we received, many of the comments pertaining to the evidence review were about the evidence ratings that we gave, in particular to the evidence rating of A to both Zolgensma and Spinraza in type 1 SMA, despite Spinraza having more evidence in a randomized trial in this population. So we discussed internally about if the amount of evidence was being adequately reflected in our evidence ratings. And we decided to keep the evidence ratings as they are. And there's additional explanation of our rationale in the report. So overall, we saw with Spinraza that it demonstrated improvements in motor functioning in all populations and improvements in survival and reduced need for permanent ventilation in type 1 and pre-symptomatic SMA. Zolgensma also demonstrated benefits in survival, permanent ventilation, and motor functioning. And both interventions had few harms and were generally well tolerated. There are longer-term follow-up studies that are still ongoing, which will provide additional evidence in terms of durability of effects and potential harms. And so with that, I will open the floor to questions.
Thank you, Alex. I'd like to ask whether our clinical experts or patient advocates have any questions regarding your presentation. All right, seeing none, I actually have a question for you. As we look at the evidence ratings, you mentioned the A ratings for both drugs for type 1. We have a B+ evidence ratings, a rating for pre-symptomatic SMA, and that's with, and confirm this please, that there are no published studies yet from the NURTURE trial, which pertains to that particular population. So could you just talk about how we got to a B+ evidence ratings with no published data?
Sure, yeah. So the interim results that we have right now are very promising. So the children are all doing very well, but again, it's just based on interim results in conference abstracts and no publications, as you mentioned. So the results are promising, but we still have some uncertainty and not enough to bump it to an A.
Okay.
I have one question. On the Zolgensma trial, you mentioned that some of the patients are receiving combination therapy or other combinations of therapy. Do we have any idea? I realize we're dealing with a small number, 12 infants. How many are on other therapies as well?
Yes. So in the Zolgensma study, of the 12 patients that were in the high-dose cohort, I believe 5 continued. I'm looking over here. And then 2 came off of it, of Spinraza.
Jean? Oh, no, Martha.
For the clinical experts, I'm wondering if you could give us a little bit more sense of the clinical
So do you
salience.
mind if I just clarify
Sorry.
one important point?
No, excuse me, please.
Pardon me.
Excuse me.
Sorry.
We'll proceed, and then we'll get to any points. Thank you.
So the clinical significance, I know has been alluded to of an increase of 3 points on the scale that's being used in the Spinraza type 2-3. Can you talk a little bit about what give us an example of what that looks like?
So the Hammersmith scale, it's a scale that's usually administered by physical therapies. It's kind of complex, so it requires some serious training. But basically look at motor skills, like, rolling, sitting, and other pretty, important motor skill, but it's only motor function. And so when that scale was actually studied and, at 3 points was deemed to be clinically meaningful, meaning that if you follow the natural history, you will never see a 3 or greater point improvement generally in an untreated patient. So, I would like to emphasize that, the different types, while the natural history of type 1 is extremely well described and extremely homogeneous, when you go to the type 2 and 3 natural history, the data is not so strong, but we do know that they do progress, but they do progress more slowly. And we also know that there might be maybe 1 or 2 point improvement early on in the disease, but after that you will never see a 3 point improvement in the Hammersmith. So it really depends when you catch them.
Right, but I guess I'm sort of trying to get a sense of the functionality that sort of signifies. And I know that 3 points at different states will be different, but is there any more you can tell us about that?
Right. So, one thing to realize about the scale is that it's looking at a variety of different motor functions. Each of them is scored equivalently, right? And you can score 0, 1, or 2 for most of them. If they can be done on both sides, we test both sides. If it's something midline, you just score it once. So you would get as much, of an increase in points, suddenly being able to walk, and someone who couldn't do that before, as someone who can just start to lift their head. So you're right, it's a 3 points improvement for one patient may be very different from another's. And the scale is the best we had at the time for doing the clinical trial, but each individual patient needs to be assessed for how much the treatment has really meant for them in their actual independence.
Thanks. David?
Thanks. I just wanted to comment on the combination therapy question. The data you're seeing at 24 months, none of those patients were on combination therapy. So the results you're seeing aren't influenced by Spinraza coming into play. you will see it in the economic analysis that the fact that we saw patients getting Spinraza does influence what we viewed as the costs. But those outcomes that you're seeing were, at 24 months, were all just on Zolgensma.
Jason.
Could I just ask, really for everyone, a question about, I'm really impressed about some of the design of these clinical trials. I mean, I think not only randomization, but also with a sham control, and just in terms of the weight, and also the very large effect estimates that we see in the endpoints. All of this is very impressive in terms of our ability to weigh evidence, at least for those populations. It also is, I think, worth noting that another thing is, I mean, imagine how hard it is for the patients and their families, right, enrolling in a trial that they know they may be assigned to sham. You know, that's probably a much harder ask. And so just to testify that for a moment. And then I think 2 of these trials, if I've got this right, Cherish and Endear, I think if I've got this correctly, were terminated early by the DSMB. So could you just speak a little bit to the extent to which we should consider that there may be biased effect estimates from trials stopped early, or may not be? I mean, it's such a large effect. It's so impressive. The design was so impressive. But I'd like to maybe any thoughts about the early termination.
Yeah, exactly. I think you've kind of highlighted the points. There may or may not be, but the effects were so large at those interim analyses that the magnitude is very promising. So even if there is a bias effect estimate, it's not going to be biased so much that there is no effect.
To alter that estimate.
Right.
Okay, thank you. We'll now transition to the cost effectiveness portion of the presentation. I'd like to introduce Matt Stevenson.
Rob, I actually just had a question.
Go ahead. While we're
comment for
transitioning,
you
go ahead.
guys. Sorry. Just regarding the safety, particularly of the gene therapy, obviously that's what everyone was worried about after Jesse Gelsinger and the OTC treatment. When I start seeing bumps in liver enzymes and only 12 patients, I start to get very worried about what the long-term safety of gene therapy is going to be. And I don't know if there's any more follow-up information or thoughts that the experts may have on that.
I just want to comment and say that as far as I know, all the bump in the liver enzyme remain completely asymptomatic clinically. They did not affect bilirubin, and they did resolve with prednisone treatment in all of the cases as far as I know. So.
Thank you. Matt.
Great. So, as introduced, I'm Matt Stevenson. I'm a professor of health technology assessment at the University of Sheffield. And with me doing the modelling are Praveen and Varun. We don't have any conflicts of interest to declare, but for completeness, I was on the NICE committee that assessed Spinraza, and my evidence group, the one that I'm director of, is the one that has appraised it for that committee. So I know quite a few things that has been put forward to NICE but hasn't been put forward to ICER. So what are we trying to do? We're trying to evaluate the cost effectiveness and the clinical effectiveness of the 2 drugs in comparison to best supportive care for SMA. So that's the objective of the mathematical model. Obviously, there is a big report out there that you can look through, but we haven't got time now. This is just going through all the methods in brief. We built a new model. It was 2 phases. The first phase is the trial data, the study data. Beyond that, we don't really know what's happening, but we do need to do extrapolations. We need to estimate what happens to the patients through their lifetime. So we did this with a long-term phase that was extrapolation. It's set in the United States. We're using a healthcare sector perspective of direct medical care and the drug costs with a lifetime horizon. 3% discounting for both costs and benefits. And our primary outcomes, there's 2 here. There's the cost per QALY and the cost per life year gained. So this allows us to insights as what happens if we remove the low utility for these patients. So the model's based on these 3 things: broad motor function milestones, mortality, and permanent ventilation. So those are going to be driving this model. For Zolgensma, it's a single-arm study. It's not controlled, so we've had to make a comparison using the sham control arm of Endear. This means that we can't directly compare the drugs, but it's the best we can do with the lack of comparative data. So here's the model schematic. This is for type 1 and pre-symptomatic. So all the patients enter in the not sitting phase. From that, we do have the short-term study data. We've got the end of follow-up, but we've also got interim points as we go through. Some of this is academic in confidence data supplied by the drug manufacturers. But you can see here the key milestones: permanent ventilation, not sitting, sitting, walking, and death. So these will move through at the time points that we have until the end of study follow-up. Beyond that, it's unclear what's going to happen. So what we've decided is that you stay in those milestones until death, with a couple of exceptions. One exception being that if you were not sitting, you could move into a permanent ventilation state. The other is, as we've just discussed, what should we do about the Zolgensma patients who got Spinraza? But I'll come on to that later. With Spinraza, at 24 months, if you were still in the non-sitting or permanent ventilation state, we assume treatment was stopped. The model schematic for type 2 and 3 is similar, but because these patients are already sitting, it's less health states. Sitting, walking, death. We have the interim points from the trial, we have the end of study follow-up, and exactly the same rules apply. So here's the data that populated the short-term model. So for SMA type 1, you can see from Spinraza we've got Endear and Shine. Shine's coloured red, as are some of the others, because it's non-comparative data. It's not controlled. For best supportive care, that was part of the Endear study. For Zolgensma, we have 3 non-comparative trials. In later onset, we only have the data for Spinraza and best supportive care that came from Cherish. And for the pre-symptomatics, we have the Nurture trial, again, non-controlled. So what did we decide to do, as already been raised, about the patients who were on Zolgensma who went on to get Spinraza? As stated, there was 5 of 12, but 2 pulled out of these, so we really think there might only be 3 that were ongoing. There were 2 potential reasons that sprang to mind, and we discussed this with the clinical expert. The 2 reasons are, 1, that the patient's health was deteriorating, so they tried to intervene with Spinraza, or 2, their health wasn't deteriorating but wasn't improving as much as parents would have liked, so they moved on to a second treatment. Both of these seem plausible, so we decided it was reasonable to assume that it was 50-50 between the 2 reasons. So of the 3 patients that we assume were in the sitting state, because that was 9 of the 12, that would say that 1/6 of the patients who had got Zolgensma would transit from the sitting state to the non-sitting state. So that's 3/9 times 50%. And that's because in this analysis, we're assuming they do not get Spinraza. So here are the survival curves. So moving left to right from here, we have the first one is ventilation-free survival on best supportive care. We've got this data. It's the control arm from Endear. The next one moving out from the origin is the overall survival if you're not sitting on best supportive care. Again, that comes from Endear, the sham control arm. One of the things that we did find when reviewing the evidence was that people on permanent ventilation appeared to survive longer than people not on permanent ventilation. We discussed this with groups such as Cure SMA. They believed that was plausible for patients on best supportive care. What they didn't believe was plausible was that same impact and effect would be seen on Zolgensma or Spinraza. So what we did there was to assume that all patients on the 2 active drugs would have the survival of permanent ventilation if they were not sitting. So those are the 3 that are quite close to the origin. You then see the gains that these drugs can promise. So the yellow line is survival in the sitting state. This is assuming that the patients are the same as type 2 SMA. It comes from the Zeres and Schonenberg paper. And the one that's really out on the far side is for those patients who walk. Here we assumed it was the same survival as the general population in the US. We did sensitivity around all of this, but I'm just telling you what forms the base case because I'm time limited. So that's the modelling methodology. We now come to try and populate utilities and costs. Utilities, and this has been quite an issue at NICE as well, a lot of them don't have face validity. What we decided to use for best supportive care is this second column. So whether you're in permanent ventilation or whether you're not sitting, we use 0.19. That was reported by Thompson et al., but came from Lopez Bastida. If you were sitting, this came from a clinician estimate for the NICE process. It decided that 0.6 was far better than the data out there. It had more face validity. So that was from clinicians, whereas the Thompson is from the carers. So these are utilities by proxies. However, we're aware these are very broad health states. There's things that might be very important to patients that we can't capture within these states. The modelling would be too complex and the data doesn't exist. So what we did was add on benefit for being on treatment for these unknown quantities. We added on 0.1 if you were not sitting, but you're either on Spinraza or Zolgensma. And we added on 0.05 if you were sitting. So there we tried to capture some of the benefits that couldn't be easily modelled. Here are the actual costs. The second column is the drug costs. So for Spinraza, it was $127,500. This came from an average wholesale price of $150,000 minus 15%. Zolgensma, we don't know the price yet, which causes a bit of a problem. We've used a placeholder of 2 million. We could use any other value and get results, but that's the one we decided was most plausible here. On top of these costs, there are, each time you get the treatment, other costs. So Spinraza, it's $1,200. Zolgensma, about $140. You can see they're listed on screen. I won't read them through. You can see what they include, infusions, monitorings, and so on. But the condition itself comes with a lot of costs. So what you have are the 4 broad health states: permanent ventilation, not sitting, sitting and walking. And the bottom line, the total monthly costs, you can see are high for the first 2. Permanent ventilation, $28,000, not sitting, $25,000. But if you can get a patient to sitting, it's believed the costs will fall to about $6,000, and walking, $2,500. You can see it's the hospitalizations in those first 2 states that are driving those costs. So that's population. So we're now going to come on to the results. We present these in terms of cost per quality and cost per life year gained, the 2 metrics that might help you in making any decisions, and also selected scenario analyses. Those that didn't really change the cost per qualities or cost per life year gained, I've omitted. We're using the healthcare sector perspective because the modified societal perspective results were similar. We're not including caregiver burden. Now, this is quite key. We're not including it because there's uncertainty around it. There's uncertainty about how we actually populate it, the caregiver burdens of drugs that are displaced, but also there's the potential that a family overall quality of life would increase with a child's death. This isn't something that we feel should be presented here. So that's why we haven't followed that. So here are the actual results. So for type 1, you can see both Spinraza and Zolgensma. Spinraza has a cost per quality of over $1 million at its price. Cost per life year gained approaching $600,000. Zolgensma, again, you take these results with a pinch of salt because we don't know the price, but you can roughly calculate it yourself with other values. Using 2 million, it's a cost per quality gained approaching $250,000 and cost per life year gained of about 180,000. The scenario analyses, these are there in case anyone wants me to refer to them later. I'm not going to go through them. These only are there if they affect the ICER, the cost per quality gain, the cost per life year by $100,000. But the key thing is that in cost per quality terms, it never fell below $800,000 for Spinraza. Cost per life years never fell below $400,000 for Spinraza. Here's the scenario analysis for Zolgensma. You can see there's a bigger range there, and it does fall, it is beginning to fall, but it's never below 150 for cost per quality or 125 for cost per life year gained. And again, this is at a price of $2 million. If it's higher, these numbers go up. If it's lower, the numbers come down. We did probabilistic sensitivity analysis, as you should do in models. What it showed is that the probability of Spinraza being cost effective was estimated to be 0%, going up to thresholds of $500,000. It's a different view here for Zolgensma, but again, at the placeholder price. What you can see there is the numbers are increasing, and at values above $300,000 per quality, all of the iterations were cost effective. We're looking at the later onsets now, type 2, type 3. It's only Spinraza here. In terms of the cost per quality gained, it was over $8 million. In terms of the cost per life year gain, because the model wasn't showing life year gain benefit in this group, it was dominated, which is shorthand for no gain in life years and extra cost. But we looked at it pre-symptomatically. This might be the best place for Spinraza to be used. So we did this based on the relative prevalence that's seen in the real world, and modelling that through, we showed that the cost per quality gain for Spinraza was now down to but over $700,000 cost per quality and $650,000 cost per life year gained. We've also modelled a hypothetical drug, because this drug does not exist yet, but it might be that there's a push to use Zolgensma pre-symptomatically. So we've used a cost of $2 million, but we've used the Spinraza efficacy. So there's plenty of uncertainty there. This is just a what-if scenario to be thought about. There, the cost per quality gains 150,000-ish for both cost per quality gain and cost per life year gained. We've done the scenario analyses here for Spinraza. We didn't do it for Zolgensma. They don't fall below 600,000 for either of the two metrics. And again, it's a poor picture. It wasn't cost effective at any PSA iteration up to $500,000 per quality. So those are the results. We're moving on to the summary now. So, model limitations. And no one's built a good model for this yet because the data isn't there, none that we can robustly believe. So there are no long-term data outcomes. There's these broad health states which could miss the benefits that we see, head rolls, finger movements. We've tried to incorporate that, but it's uncertain what they are and how they're valued. There's a lot within the main report, there's a lot of sensitivity and scenario analyses that try and address these concerns. So we first put out the draft report, put it out to the public, got your comments back, and this is how we acted on the key ones. The first was that it seemed to be lacking face validity, that if you're on Spinraza or Zolgensma, that once you have permanent ventilation, you'd live longer. That came through loud and clear from Cure SMA and other groups, so we changed that to one that they believe was reasonable on the phone call. The fact that the health states may be too granular, again, that's true. They might be too granular, but the data doesn't exist to do anything better. And we included this utility benefit. The third one was that we hadn't calculated the proportion of patients sitting in the Spinraza arm. I think that was true. We acknowledged that it was a mistake and we changed those numbers. But those are the three key things that came through. So, in terms of the summary of the cost effectiveness, at its current price, Spinraza was not seen to be cost effective, given the standard thresholds. There were over 700,000 in terms of cost per quality gained in the pre-symptomatic and 650,000 in the life year gained. Zolgensma, again using this 2 million potentially fictitious price, the ICERs are still high, cost per quality gain over $240,000 and life year gained over $180,000. Finally, there was a paper published recently that might be brought up and you might want to ask me questions about. It was the cost effectiveness of Spinraza in Sweden. So we've looked at this and it's very similar to the model that initially came to NICE. There's different utilities in there, but it's very similar to that model. Biogen have now changed that model 3 or 4 times, depending on how you count it, and it was presented at NICE yesterday. So I can talk about that. But in terms of this actual model, the third bullet point talks about what the NICE committee felt, and that was the committee I was part of. So the reason the results might be different, the NICE committee said the transition probabilities are optimistic, do not reflect clinical practice. The long-term overall survival benefit, optimistic assumptions, highly uncertain, and that the utility values are highly uncertain. The key thing in that is that patients on Spinraza could never decrease in milestones. They stayed the same and then a proportion of the cohort got better. So you ended up with a proportion walking that was quite large, that was not being seen in any trial. The later models have ramped down all of these optimistic assumptions to one where they believe it is far more reasonable. But this paper is using the ones that have since been changed and has not been accepted by NICE. Thank you, and happy to take questions.
Okay, questions from the CEPAC panel? Steve.
Thanks for your presentation. That was terrific. A couple of questions about how you model death. So in the patients with type 2, type 3 disease, was there any consideration to modeling death from SMA and death from other causes separately? And then a question with regard to how death was parameterized. The exponential distribution was used for a number of the decisions, and I understand it was informed by model diagnostics, but was there any thought that it may be overly generous because of its non-parametric form?
Okay, the first one, no. We just modelled an overall mortality. It wasn't split. I don't think we'd have the data to do that. The second one, again, yeah, if you look at all the NICE technical support documents, you'd be looking to see a rate of change of hazard, which is what you're talking about with the exponential. So for those who aren't naturally statistics, the exponential assumes a constant rate across time. So it's always the same proportion. Here, though, because of the model diagnostics, the AIC, BIC, there was no strong urge to use a different one. If we'd have fitted, say, a Weibull or a log logistic through that, I'd have expected you to be asking, well, where did you parameterize that? Which clinicians gave you the change in hazard? So it may not be an exponential, as in with all of these things, like the CAR-Ts you may have looked at. There's lots of things like this we talk about. So I'm comfortable with what we've used, but you're correct, it might not be an exponential.
Chris.
Thank you for that very clear overview. Question to you or to Dr. Ellis on the reasons for discontinuation in any of the trials. If they looked positive, was that a crossover from sham to active comparator, or were they just discontinued for a whole host of other reasons?
That'd be you, Alex.
Yeah, so discontinuations from the trial for types 1 and for types 1. So for the Spinraza arm, the discontinuations were leading to adverse events that had led to death, but they were not considered treatment related. So I think in the modeling, that's not something that's explicitly being captured.
How were, I just know from the Thank you for all this wonderful
Oh,
work.
yeah,
Yeah, I just saw in the report that the prior authorization policies in the United States in some cases involve confirmation by genetic testing. Was that, I guess my first question is, was that, I'm not actually sure how one would consider that in the model, because you have to say if you would send the, are there situations where you would send the, this is sort of becoming a clinical expert question, but you would send the test if the prior authorization requires it, but you would otherwise treat without it? Was that considered in the model, and does this matter in terms of the result?
It wasn't considered in the model. I'm not sure I fully understood what you were asking, though.
Yeah.
But in terms of would you need to send it off to get confirmation before treatment, wasn't considered in the model. It would be assumed, though, that the clinical practice would be responsible, and that would happen. So it was sort of like implicitly there, but we didn't consider treating people who didn't have the disease. Is that the question?
Yeah, I guess I think because of the dollars involved, it presumably wouldn't change it. But I guess maybe it's becoming more of a clinical expert question about, like, are there situations where, like with familial hyperlipidemia, there's situations where you sense that they have it because of the numbers, but you have to send the test simply for the prior authorization. Is that ever an issue?
I think that in clinical practice, you always confirm the diagnosis of SMA with genetic testing. But in the prevalent cases, if you're trying to get pre-authorization, we had situation where we needed to repeat the test because the older test will not report the number of copies of SMN2, and that was a requirement in many policies.
Got it.
So it will be a second test that otherwise you wouldn't have repeated.
Got it, thank you.
Hey, Martha.
Thank you very much. Couple of questions. The difference between the life years gained in Spinraza versus Zolgensma is quite significant, and I'm just trying to have a sense of why that disparity.
Okay, that one's quite easy, actually. If you remember the curves that I put up there, the walking was thrown right out. I wrote these down in case somebody asked you. But the walking life expectancy was 79 years, sitting 29 years, 5 years for non-sitting.
Okay.
Now, if you end up, as we use from the short-term data, if you end up in Spinraza having more people not sitting and sitting, and in the other drug you have more people walking and sitting, you end up with a higher life expectancy, and that's why there's a higher life years gained.
Great. And then the second question is just about the difference in treatment costs for Spinraza for the pre-symptomatic versus the type 1. That seems like a huge discrepancy in drug treatment costs.
Okay, I didn't expect that one. So in here you're saying that the 10 million is a big discrepancy with...
Versus 2 million in the infantile onset.
It's similar. They're walking. So they live longer and it's constant treatment. So you're racking up the costs as the patient lives.
Okay, thank you.
Okay.
So I also want to ask our clinical experts or patient representatives if they have any questions or comments. Okay. Any other comments from CEPAC members or our ICER research staff? Nope.
Can I ask?
Yeah, one more.
Could you just speak briefly to the willingness to pay threshold that's appropriate, generally considered for rare diseases? Because I think the paper that you referenced used a willingness to pay threshold. It was a Swedish analysis, as you noted. I looked it up. It was just under $200,000. And I know you did different analyses at different willingness to pay thresholds, but what should we consider as an appropriate willingness to pay threshold for a rare disease?
Well, I can tell you exactly what NICE do, but that's probably not what you want to need. I'll pass that one to Steve.
How about we hold that before we take the votes? We can bring that up as a broader issue.
All right, we'll now transition to the manufacturer comments. I'd like to invite our manufacturer representatives, Douglas Sproul and Jonathan Young, to the front table, please.